Ladies and gentlemen, welcome and thank you for joining Invasive Species webinar series. Before we begin, please ensure you have opened the WebEx chat panel by using the associated icon located at the bottom of your screen. If you require technical assistance, please send a chat to the event producer. Please note that all lines are muted until the Q&A portion of the call. We will give you instructions on how to ask a question at that time. You may submit a written question at any time by selecting all panelists from the drop-down menu in the chat panel, enter your question in the message box, and send. For optimal viewing, we recommend side-by-side view. Move your mouse to the upper right corner of the whiteboard next to the video strip. A small white circle with black boxes will appear. Hover over that circle and select side-by-side from the three presented options. As a reminder, this conference is being recorded. With that, I will turn the call over to Courtney Chambers. Courtney, please go ahead.
All right, thank you, Luke. Hello, everyone. I'm Courtney Chambers from the Engineer Research and Development Center. I'm glad you could join me as we continue our Invasive Species Fall webinar series brought to you by the Corps of Engineers Invasive Species Leadership Team, partnered with the Aquatic Plant Management Society. Throughout our fall series, we are summarizing the latest research and technical information on management strategies and a variety of invasive management strategies for a variety of invasive aquatic plants and harmful algal blooms. These topics are presented weekly on Wednesdays at 12 o'clock Central Time, just as we're gathered here today. This week, we're focusing on giant Salvinia and other floating weeds with presentations by 3 experts. Each presentation will be approximately 20 minutes, and then we'll have a question and answer session for those able to stay. We'll get to hear about chemical control of giant Salvinia, those updates and challenges, giant Salvinia about control agent rearing, release, and field establishment, and then also updates on biological control of giant Salvinia from LSU. All right. Oh, I'm sorry. One more minute, please. Okay, a few more notes before we get started. We, as Luke mentioned, we are going to be utilizing the chat box, and that's how we want you to submit your questions throughout the presentation to all panelists. And then during the question and answer portion, you'll also be given instructions to ask questions verbally. So we definitely want to hear from you. In the upper right-hand corner, like you mentioned, you can select that side-by-side view for optimal video display of our speaker. And then our webinar is being recorded, and I'll be sure to share a link to the presentation files following our meeting. Okay, now at this time, I would like to introduce our speakers today. Dr. Chris Mudge is a research biologist for the U.S. Army Corps of Engineers Research and Development Center and is stationed in Baton Rouge, Louisiana. His current research responsibilities include the evaluation and development of use patterns for recent registered and experimental herbicides for selective management of invasive aquatic plants that hinder our nation's waterways. In 2007, Dr. Mudge joined the Chemical Control and Physiological Processes team at Burdick in Vicksburg, Mississippi, and then in 2013, he was reassigned to Baton Rouge, where he's an adjunct professor at LSU in the School of Plant, Environmental, and Soil Sciences, and serves as an advisor to master's and PhD graduate students. He currently serves as a member of the board of directors for the Aquatic Plant Management Society, as well as a committee member and or a chair of numerous APMS, Mid-South, APMS, and Weed Science Society of America committees. Okay, and then we'll have Ms. Julie Natchery, and she's a research biologist for the Environmental Laboratory of the U.S. Army Engineer Research and Development Center, and is stationed in Texas at the Louisville Aquatic Ecosystem Restoration Research Facility. Julie's work primarily focuses on biological control and use of host-specific natural enemies to manage invasive aquatic plants. But Julie has been involved with invasive plant management for 18 years, with work spanning from east to west coast of the United States and a variety of plant species, including giant and common Salvinia, hydrilla, water lettuce, water hyacinth, water primrose, Eurasian water milfoil, and alligator weed. Her research interests include novel mass rearing and monitoring methods for biological control agents and the effect of plant nutrition on the efficacy of biological control agents. And then our third presentation is by Dr. Rodrigo Diaz, who is an associate professor in the Department of Entomology at Louisiana State University. Dr. Diaz's research focuses on biological control of non-native weeds and insects. His current projects include biological control of giant Salvinia, roseo cane scale, elephant ear, and air potato. Rodrigo obtained his master's degree at Texas A&M in 2003 and his PhD at the University of Florida in 2008. Thank each of you for bringing your knowledge of aquatic plants to us today. And at this time, Dr. March, we can share your slides and you can begin.
Okay. All right.
Okay.
Okay, thanks, Courtney. Okay, good afternoon, everyone. So we're going to talk about giant Salvinia, the floating fern that's been here for approximately 2 decades, invaded Louisiana, Texas, and spread throughout the rest of southeast United States, and we found in many other states, and a lot of populations here and there have been eradicated, but it still is a major nuisance for us locally down here. As I said, it's a floating, it's known for its fronds, that's essentially the leaves on top, and it has submerged root-like structures, which we also call submerged fronds. But what is really detrimental is because of its exponential growth rate. It can double in coverage in as little as 36 hours up to 53 hours. So it can take over a water body very quickly across the surface, and then eventually you see it matting up to maybe even a meter thick, at least 2 to 3, 4, 5 plus layers thick later in the growing season. So it becomes detrimental to all the wildlife and fisheries habitats. So as I said, it started off in Louisiana. Besides the initial population that was found in South Carolina that was initially eradicated, but Toledo Bend Reservoir, which sits on the Louisiana and Texas borders, where everything's spread over in West Texas, a lot of the core reservoirs and other state lakes, into pretty much every water body in Louisiana, both natural and man-made. We're talking about Lake Pontchartrain, the coastal marshes, even at times Mississippi River, and so forth. And then it's spread along the Gulf Coast in places like Ross Barnett Reservoir in Jackson, as well as the Lake Marion and Lake Moultrie system of Santee Cooper, which I'll talk about later on. So there's many challenges, just besides the difficult to control weed, but one of the biggest problems we'll have, and it's probably with any form of invasive plant management, is the budgets. We see yearly cuts. In particular here, because of COVID, in places like Louisiana, the budget has been cut by more. Once you get beyond that, we have 15 aquatic herbicides registered for aquatic use, but about 10 work to some effectiveness, and then the 5 are really good. But we could always use more anytime you have an invasive weed like this. We also see a place like Louisiana, there's 17% of the area is made up of water. So you have these continuous nutrient-rich systems that connect that are low pH, that the plants thrive. And there's a lot of heavily forested man-made water bodies that were made through impounded swamps. And when you have a lot of trees, it's hard to get to manage the plant, not only from the herbicide standpoint, but even releasing the weevils, and definitely not with mechanical harvesting. In addition, one of our biggest things that we're just far enough south is that the winter conditions in the south really don't knock the plant back enough to have a whole lot of die-off. And then up north, where we would see on good years where we have a cold winter, we do unfortunately have the weevil mortality, which my other speakers today are going to talk more about. So you kind of lose your biological control arsenal right there every so often. And, of course, public perception. Regardless if you're using the herbicides or the weevils or mechanical, there's always that you're doing too little, you're not spending enough money, or why are we spending all this money? You should focus on one technology to others. So there's always the backlash. You can't win either way when you still have that weed after 2 decades. So, sorry, this screen's not advancing. Okay, there we go. When we talk about just herbicides, which is my description of the presentation, we have kind of 2 general seasons. Most of it is the growing season, where we're talking, March, April to October, November, and we're doing a lot of foliar applications, spraying herbicides onto the foliage of the plant. And a lot of it focuses around glyphosate and our combinations with things like diquat, flumioxazin, or carfentrazone. And then the latest trend has been using pinoxalam in combination with 2 of those herbicides to kind of mix it up. And then we have a recent herbicide that was registered, which I'll talk about later, with metsulfuron. And then on the flip side, we can do subsurface applications where the herbicide is put into the water column with pinoxidine and fluoridone. And then the other end of the equation would certainly be winter applications, which is another part of my talk, but that's when the plants are slower growing, and that's a more foliar application with dicot or flumioxazine. So aquatic herbicides, when they're sprayed to the foliage out of a boat or an aircraft or whatnot, you can see anywhere from slow to rapid control when you get good contact, and it's going to work on the plant. But we do see failures when we're talking about plants that are matted up, several layers thick, because whatever you don't contact, the herbicide is obviously not working. So you may need repeat applications or get into integrated pest management. Now, if you go with a subsurface application, you do have a much slower control. All the plants are exposed, and essentially they are bathing in the solution of the herbicide for a number of weeks if possible. But the unfortunate part is that these large systems require a lot of product, and you could need anywhere from 8 to 12 weeks, probably an average of about 10 weeks of exposure to the product. So that means repeat applications oftentimes, and it can become cost prohibitive in larger systems. But in smaller ones, it has shown some effectiveness. So when we talk about weed management, after I moved down to Baton Rouge in 2013, started looking at the things that they were doing, and they were having some success. And I say they meaning the agencies in Texas and Louisiana where the primary populations were at, but we needed to look at new alternative ways. And one of those ways was, how do we look at wintertime management? And that's a time that most managers and crews are doing downtime, especially in Louisiana and Texas. It's hunting season, or it's off time, or fishing or whatnot, and they don't want to be out on the boat or it's cold. But in places like Florida, primarily, we've seen success where there is maintenance control of water hyacinth. And of course, part of that is because you do have milder winters, or in a lot of cases, south of Gainesville, really Orlando, where winters are almost non-existent. So the plants are growing year-round. But up here, we do see some browning, and plants may fall out of the water column a little bit, and they're difficult to get to. So when you look at these hardy zones, it's kind of where do we see these temperature regimes and these breaks across the state? And really, you can see in the wintertime 5 to maybe 10 degrees different at one point in a given day that north or south of Louisiana. You wouldn't think, but there is. And the same thing to me as you go into Mississippi and Texas the other way. And so there will be minimal differences down south as you start to get to the central part of the state around Alexandria, definitely, and then up to Shreveport and the top, you'll see changes. So that may have an impact on the plants as far as injuring them just from Mother Nature taking care of some of our problems. And if you see across the United States, looking at the USDA map, you can see that similar trend just going right across the central part of the deep south. And if you look at the pictures out at the bottom, there's a giant Salvinia in a trash can, and the far left one was a thin layer. Well, and that's in Baton Rouge when we had an ice period where it was below freezing for a few hours, and it had about 1 to 2 inches of ice. The plants died. And now you start to look in the central part, there's thicker layers. And on the right two photos, we have frost, or, not even freeze damage. The plants on the top were burned. You pull back those top layers, and you can see there's healthy plant material below. So it takes many, many freeze events in order just to knock the plants back, especially if it gets layers and layers thick, if no management is being done. So if we show up north where it's a little more severe, we can see more ice periods and frost and more frequent events. So this does help out a lot compared to down south, because the Salvinia compared to the weevil, just from the natural populations that have been brought into Louisiana and Texas, there is a little bit more tolerance for the cold as far as the plants are concerned than the weevils. We've seen some natural adaptations, and Julie and Rodrigo are going to talk about that. But, but originally was the plant could survive at lower temperatures for longer periods of time. That made it difficult. And versus, where we're looking down south. I have a photo here showed in Chalmette. This is just outside of New Orleans. And you can see where I was with the LDWF with Jonathan Winslow a few years ago, and we were just looking at sites and seeing what had happened just riding around. And we went up on a levee, and I took this photo, and I said, wow, these plants are going to die, and we see some ice on top and good damage. And I happened to come back 6 months later and looked around, and I know it was in the same spot because I remember those power lines, but I saw that boat on the right, and that was from Katrina. And that's what Jonathan had pointed out, that it had been, put in that spot and flipped over. So I was just a few inches away from where I was previously, but look how healthy this plant is. And you can see, it's a little bit damaged. You can see the browning in the foreground, there's the weevils were feeding right there. But with a mild winter, 1 day of freeze, it didn't do anything. And that's, some of the challenges. But we know that it offers some level of activity, just Mother Nature. So if you look at a wintertime scenario where it's more of a kind of a normal, colder winter, and the plants are knocked back and you have a single layer, and you were to go in with a herbicide application, as in the top upper right-hand corner, you can see the plants as you go down to below, they're brown. The application worked, and you go to the next photo over the bottom left, the plants were pretty much gone. There were some random plants here and there, but it was great because you had a single layer come out of the winter. Let's now go flip the script with a mild or non-existent winter where the plants are not really, knocked back. You're not doing much management late in the year, and you see if you plant layer 6. So if you were to go and spray these herbicides to the foliage, you do get the top level burn, but then everything else comes back and you have more plants there. So just by Mother Nature not being as cold could cause some issues right there. Staying on top of it is beneficial. And in addition to that, twofold. Whether you're talking about a winter or not, you do have hiding places. These plants can get out in hollowed out trees and logs, and then even up in the foliage and the vegetation and trees where you can't reach with the herbicides. And, we're talking even where you have vegetation, both like water hyacinth or some native species covering up. So not only the foliar sprays, but the temperature is a little bit warmer down there, so maybe the frost and freeze won't impact it as well. So a lot of different things can kind of compound the issue. So basic question was, can we get control? What happens if we apply late in the year under cool or cold conditions? What's the plant response going to be like, and does it matter what type of herbicide? So we did some trials in 2014 and 2015, and some of these products, we've changed up the mixes a little bit. So, you have to think this is 6 years ago, some of this. And we went into standard treatments that have been used in the state, and a lot of folks are on glyphosate and dicot. But the point of the whole study was, regardless of the product, was to look at it in a couple different scenarios. The first was we had a cold winter coming up, so I set the trial up, and I wanted to look under severe conditions where the plants were not covered and you had an open water freeze event. This is South Louisiana, Baton Rouge, so we were cold enough. And in the following year, we were predicted to have a mild winter. The plan was to repeat the original study, but when things weren't as cool, based on the predictions, I said, well, let me just see what happened under, almost a non-existent winter. And I set up 2 trials where we had open water, where we allowed the natural frost to occur, and then the other where we had a canopy protecting it. And what we saw was, you look at the first trial under uncovered severe winter conditions, and even you look at the control and around the red boxes on the left side, we can see as we go through time, 1 week to 2 to 7 weeks after treatment, the plants were knocked back. And then when you switch over to the other 2 treatments, we could see we'd sprayed these, and about 2 weeks after treatment, we had some frost and freezes, and the plants were knocked back severely. And that was reflective in the biomass data whenever we concluded the trial and harvested the material, looked at the data loggers, and had 32 hours at freezing or below for 5 hours, right about 2 weeks after treatment, mild injury, and then the herbicide on, plus the freezing event, substantially knocked back the material. And then you can see 99.7 to 100% control. So really great. So the herbicides had an impact. I mean, there's still, if you didn't manage, you would have giant Salvinia, and this would be a source of populating for the next year. But the herbicides kind of kick-started everything late in the year. And this was in approximately December when they were sprayed. And this is published in the Journal of Aquatic Plant Management. You can look under my name and Bradley Sarton. So the following follow-up trials, we're looking at mild winter conditions. And what I did here was, as I said earlier, excuse me, we're going to go uncovered versus covered. Look at how much material is there on the control tanks, a substantial amount. And the only difference there was I put a tarp over it. Anytime we saw a freeze or frost coming, I just covered the tanks just to make sure there was no damage from that. And even looking at the herbicides in the middle and the right-hand pictures, you can see there's more biomass, and that was definitely reflective. At the 15-week mark when I harvested all the material and dried the plants, all treatments worked regardless of trial, but look how much better control you had under uncovered conditions. Now, we still have 72% of the materials there and 99. None of the treatments worked completely, so it was always biomass. And then whenever it was covered, so we allowed really no winter influence, the herbicides were less effective. Or really, I shouldn't say the herbicides were less effective. They have less activity, and we didn't have the cold weather kind of pushing it along and helping them out. And then nothing, it did tell you that dicot, which is our winter treatment, was the most effective, and that's a contact herbicide that works quickly. So that kind of was, into that game, and I want to look at something, the new one, and I had a new graduate student at the time, Dr. Bradley Sarton, who works with me now at ERDIC. Part of his many chapters in dissertation research was to screen herbicides. And he looked at 12 non-aquatic products that were registered in anything but aquatics and wanted to see for activity. And through all the screen process, he found a couple of those. And that would be sulfometron and metsulfuron, because they provide excellent control. And we started looking at the tox packages, the half-lives, and other environmental impacts. And we found metsulfuron had some good characteristics. So then we had a couple companies came forth and approached us about looking at a special local needs, and two herbicides with 4 special local needs were granted over the past year and a half in Louisiana, Texas, and South Carolina. And we were looking at public water use only by no private applicators unless you're contracted, but mainly one of the state and federal agencies applying this at a half an ounce to 1 ounce per year. Now, these numbers, if you look at it, half an ounce to an ounce is very, very low. Most of our products are used probably at a minimum of about 4 ounces, and a large majority of them are used a lot higher than that. So it is a change in much lower application rate. So looking at my former student, Will Prevost, who did his thesis research, finished up about a year ago almost, and he was looking at a rate titration trial. So he took Bradley's information and looked at how low could he go, and he evaluated a 1/16 of an ounce all up to 4 ounces. And you can see this product alone was slow after 2 weeks, but there was a good bit of browning. And looking at the 2 lowest rates, started to see a little recovery at 2 weeks. And if you switch over then to the, and I apologize, should be 8 weeks after treatment, but so this was 6 more weeks later. The only tanks that had any plants in there were the lowest rate, and then the next one, the 1/8 of an ounce. But everything else, we saw really great control. And when he analyzed the data, did a nonlinear regression, looked at steep, sharp curve, basically says that plants were controlled at substantially low rates and 94 to 100% control. And when it calculated LD90, or the lethal dose, to kill 90% of the test population, he found that it was less than a tenth of an ounce. Now, don't get me wrong, it didn't kill it that low, but it showed it had, great activity there. So, great product. This information will be published in the first issue of 2021 in the Journal of Aquatic Plant Management. Both, we've got 2 publications come out that you can see this information, as well as his dissertation online. In the follow-up trial was look at compatibility, knowing that this product is very slow. When applied to giant Salvinia, it was doing the job, but it took a while to really take the plant down. So he tank mixed it with glyphosate, diquat, flumioxazin, or fentrozone, and found really great results, and showed that everything, when it was tank mixed or alone, provided 98 to 100% control. So very effective, compatible, and it was slow alone, but it did eventually do its job, and that was also part of the published work. So that was our new herbicide, and we're still continuing to look for products. The next few slides, I'm going to talk about a couple specific sites. So all the hard work that we've done, unfortunately, hasn't provided much as far as control. We're still seeing the spread of it, this invasive species. But whenever it came and found out, I was contacted by Santee Cooper from Lake Marion and Lake Moultrie, that giant Salvinia was discovered there approximately 3 years ago. There was some alarm and trying to figure out how to get rid of this. It's a big hydroelectric system just outside of Charleston. And they found 5.5 acres in 2017 and treated it, but in 2019 it was almost 600 acres. So they've had a big campaign to try to manage that species, try to get rid of it as possible. But they've got a lot of the water bodies that have the vegetation that's kind of hidden underneath natives, other invasive trees. It's a flowing water system. So other challenges compared to, similarities and differences than what's over in Louisiana and Texas. And, as I mentioned, the mixed populations, they've had water hyacinth and hydrilla for a number of years, but probably, I don't know the exact number, but it's been at least 5 or more years, maybe close to 10. They've had crested floating heart, and that highly invasive species has caused problems, both because you have mixed populations and you're trying to figure out, what works on weed A versus weed B. Some of that you can't use anymore. You have to go to mixes or other techniques and technologies. But also the crested is providing a kind of a harborage and blocking out where giant Salvinia is, just like the water hyacinth. That's a big issue. So they've tried a lot of different treatments. and they just kind of thrown the book at it as well, based on what our knowledge and our list was and what they were using before for crested floating heart. And, they've had pretty good success on a lot of these treatments. They've gone, kind of a little bit different mixes, and also recently incorporated the weevils. Rodrigo was in contact with them providing some, and then I know I've given Doodle's information. So they're going to try to hit it at all angles and see will the weevils survive, because we have to have an IPM approach here. And most of them worked. There was a couple that weren't didn't do as well, but they're still continuing to try and evaluate. One treatment they really liked was the Floridone subsurface at 30 parts per billion, and you can see that photo on the left. A lot of, Salvinia and some, duckweed in there, and probably some crested floating heart here and there. So it was mixed up, and they were seeing a lot of initial bleaching and taking the plant out. So, when you have smaller water bodies and you're not treating thousands and thousands of acres, or you have to treat the whole state, this is a great treatment. And I think it's an opportunity in places like this that you can go with a Floridone or a Panoxalam, go subsurface and treat, widespread with it and get in there. You still have to remember you've got the dilution when the water moving, and you might have to do some what we call bump treatments or reapplications every so many weeks to maintain those lethal exposures. But a good opportunity to try something like this as far as the, in-water applications. And then the final kind of part of the presentation was one of our more recent places where giant Salvinia spread. I mean, we've seen it in pockets in Florida and Alabama and Georgia and here and there, but it's either eradicated or doesn't cause much of a problem. But this is a big drinking water supply, and this was at, think about along I-20, the northern part of Louisiana as you come across the state line. That's where Ross, Barnett, and Jackson are located. So it's a big drinking water supply, and they wanted to contain it as much as possible. There was a lot of state agencies, Pearl River, Water Supply District, and then the Mississippi Department of Wildlife Fisheries, as well as Mississippi State and ERDC, were all jumping on board about, trying to make recommendations and come up with anything that maybe had worked before or new. And we're talking about anything from containment booms to aerial applications, mixes, drawdowns, surveys, desiccation, burns, relying on the weather, et cetera. They didn't want this problem to get out of hand. And they started off right away trying to put the booms out and keeping the Salvinia where it's at and put it minimal. We even see places like, the boaters are going to be spreading it. So keeping it contained and isolated is was a big challenge. And then they started the drawdown process of removing the water and isolating and keeping it, stuck in certain spots so they can manage it better. Even after they did those types of technologies and treatments, they were still seeing the giant Salvinia thriving in spots where it's kind of got a lot of other vegetation on top of it and it stays moist. Giant Salvinia is like a sponge almost. So it stays there, it's protected. So they decided, what's next? We have open water, we're walking around spraying, we're missing some of it, it's still there. So they started doing, controlled burns and trying to just get rid of whatever's there. And since these were, not near homes necessarily or a little bit away, they were able to kind of treat it in prescribed burns type applications. And then they got those areas under control, and then they were seeing places where they had a lot of buttonbush, so small trees, bushes, and they were doing aerial applications where they couldn't reach by boat, and still, unfortunately, a lot of that material was still protected. Salvinia didn't receive all the spray, and it was underneath that canopy. So they came in there and they went with the drawdown even more, and then allowed the some of it was the cut and removal, spraying and burning, so kind of a repeat of everything. Good news, according to Ryan Jones with Mississippi Department of Wildlife, was that so far the last few pieces they found of Salvinia, they were removed, and they haven't seen the Celtic. They're going to stay on top of it, both trying all these techniques of the drawdowns and the surveys, but they're also going through the education portion of it, of letting the public know in these campaigns, and they're trying to contain and treat and monitor because they want, if they can get this out of, underway, they want to keep it away. And of course, they're relying and hoping that Mother Nature will provide a freeze and get rid of some of it. Well, now they remove all that excess vegetation. With that being said, I do appreciate everyone's time, ISLET and APMS for hosting, as well as former students and colleagues at ERDC, the Ag Center, LDWF, Nancy Cooper, Mississippi Department of Wildlife Fisheries, Mississippi State for providing both either labor or these slides. And thank you.
I'm going to go ahead and
Okay. Hi, guys. So I'm going to talk about our biocontrol efforts with giant Salvinia. So just a quick background here. We have one host-specific biocontrol agent for the control of giant Salvinia, and that is the Salvinia weevil, or Sturdivagus salviniae. The adult stage feeds on the buds and leaves of the plant, which causes growth suppression. The larval stage tunnels within the rhizome, which disrupts nutrient flow and effectively kills the plant. So it's very important to notice that the larval stage of the weevil is very important and is the stage that actually kills the plant. So the Salvinia weevils have been successful at managing giant Salvinia in 13 countries worldwide, but success has remained low in many areas of the temperate regions of the U.S., and this is primarily central to North Louisiana and central to North Texas, and this is primarily due to unsuccessful weevil overwintering. So I work for ERDIC, but I am stationed at a satellite facility in Lewisville, Texas, the Lewisville Aquatic Ecosystem Research Facility, and we are about 45 minutes north of Dallas, and we have been working with Salvinia weevils and giant Salvinia since 2003, when we first began in outdoor natural half-acre ponds at our facility. But we quickly learned that just being 45 minutes north of Dallas or that far into North Texas, that we had very inconsistent overwintering with either giant Salvinia and or the weevil from year to year. So we converted over to using these above-ground culture boxes that are housed inside these cold frames. And this is a winter picture, but during summer we can take down the front wall and the back wall and roll up the side walls to create an open air, kind of natural temperature conditions. And in 2012, we substantially expanded our efforts of rearing, release, and monitoring for giant Salvinia biocontrol. We moved into working with Louisiana's Department of Wildlife and Fisheries, and the project there spans in multiple water bodies from north to south Louisiana. And we also moved into 2 Corps of Engineers reservoirs, southeast Texas, and this is Steinhagen and Rayburn. And at this point, we started doing very large-scale field releases and year-long sampling at 4 to 6 week intervals, and this is year-long, and we have been doing this since 2012. And the reason why we chose to go with this year-long sampling regime is that we knew from the get-go from past literature that was already out about the Salvinia weevils that we were probably going to have difficulty with successful overwintering during winter. So we wanted to make sure that we were closely monitoring weevil establishment success during the colder temperature months. So just a quick kind of overview of biocontrol in general, and I've kind of really, simplified this. But in general, when you work with biocontrol, your first step is to establish your insect or your biocontrol agent. And when you do this, this can be further, split into short-term and long-term establishment. So when you first go out and initiate a new biocontrol program, you're trying to release an insect into a new environment, a new water body, and you're looking for insect presence and population growth, first just during that initial growing season, but what you're really hoping for is long-term establishment over multiple growing seasons, which would indicate successful overwintering. And you really can't move into step 2, which would be using biocontrol to manage your plants unless you get successful overwintering and long-term field establishment of your biocontrol agents. And we noticed pretty early on with our work with the state of Louisiana that we had site-specific differences of variable weevil establishment and population growth. And this was both between sites on the same water body and sites on different water bodies. So we started looking into what was limiting our success, and really our first thought was temperature. We kind of knew from the very beginning that when we worked in central to North Louisiana that we needed to, pay attention to winter cold temperatures. And there's also a lot of work in the background literature about giant Salvinia about plant nutritional quality, that plants that are less nutritious, less nitrogen, that the weevils have a harder time establishing on those plants. So we, decided to kind of focus on those things. So we started in 2012 in northern Louisiana, but I'm going to show you data from 2013. And this is data for Bistineau and Caddo, and I have their lat longs here in case you want to kind of follow along with where we're at versus north and south. And both of these reservoirs are located in the Shreveport region, so North Louisiana along the I-20 corridor. And some main things to point out here. So in 2013 on Bistineau, we released 100,000 Salvinia weevils. spread amongst multiple releases from April to October. So usually about every 4 weeks, we'd go out and put another batch of weevils. At Caddo, we released approximately half as many weevils, 52,000, and we released for a shorter amount of time, April to June. So when you look at this graph here, this is weevil density. The blue bar is weevil larvae, and the orange bar is adults, and this is data from April of 13 to late February of 14. So we came in and we had basically no weevils. We started doing weevil releases every 4 weeks, and during the growing season, we started to detect, decent numbers in August here. And if you look over here at Caddo, same thing. Even by June and July, we had pretty good, medium to high weevil densities. So for this reason, we stopped releasing in Caddo in June because we could already see a good amount of weevils here and thought, it'd be best to put the available weevils in other reservoirs. But in Bistineau, we were shocked to see that when we visited the sites in August and then came back in October, that there was a 96% reduction in our weevil density. We're still in the growing season. We're actually entering kind of milder temperatures. So we really couldn't pinpoint why we would have lost these weevils at this point. And we really, thought that maybe it was a fluke. It had something to do with our sampling methodology or, basically a fluke. And so we went ahead and re-released in October, and we came back in November, and we detected a small amount of weevils here. Well, then we went into our winter time period, and the winter of 2013 to 2014 was actually a very cold winter for Louisiana. So from November to December at Bistineau, we lost 80% of the weevils, and at Caddo, we actually lost 100% of the weevils. There were no weevils detected in December. This trend continued into late February, where we detected no weevils at Bistineau or Caddo. But while I have simplified this data to just show you Bistineau and Caddo, we were actually sampling and working on multiple lakes, multiple water bodies in northern and central Louisiana, and there was no successful overwintering in any of the reservoirs during that winter. So in comparison, I've kept the Caddo graph here, and I've added Blind River. So Blind River is located in South Louisiana near Baton Rouge. So in contrast here with the Blind River site, we only released 16,000 weevils one time late in the growing season in September. And you can see that when we came back in November, we had almost 1,200 larvae per square meter of Salvinia and almost 900 adults. So vastly different establishment rates, population expansion rates when you're looking, going farther south with those more favorable conditions for the weevils. So like I said before, this was a very harsh winter with cold temperatures. So even in Baton Rouge, they had quite a few cold spells. And from November to December, we lost 50% of the weevil population. But when you're starting winter at almost 900 adults per square meter, you're in a really good position to lose half your weevils. You still have, more than enough weevils. We did continue to lose them down to a very low number in February. But the important thing here is that this was one of the coldest winters that I've experienced during this project. And at Baton Rouge, our southernmost site, the weevils were able to persist and at least overwinter to some degree. And they were able to restart their own populations the next year. So the same pattern, so the work I just showed you was from 2013, but for 5 years we saw the same pattern with Bistineau and Caddo with our two northern sites, that during the growing season we would release weevils and we would see population growth at Caddo. And at Bistineau, even during the growing season, we could not get the weevils established. We'd have very limited weevil establishment, basically just coming from us constantly putting weevils in. We weren't really seeing that they were naturally establishing or persisting on their own. And at both Bistineau and Caddo, we had difficulties in overwintering. During the 5 years of this data, starting in 2013, we had successful overwintering 1 year for Bistineau and only in two of these winters for Caddo. And, to kind of put this in perspective, so we overwintered at Caddo 2 out of 5 years, but these were not consecutive winters. So what this means is that we overwintered one winter, and then the next year the weevils did great, and then we lost everything that next winter. So for most of the years of this project, because of all the winter mortality of the insects, we were starting from scratch every year, having to reestablish weevils. So I said earlier that at Bistineau, we didn't really know what was going on and why we couldn't establish weevils during the growing season, but this pattern continued for 5 years. When we started looking at water temperature data, And like I said before, we were monitoring a lot of lakes, but we're just going to look at Bistineau right now. So by mid to late June, the water temperature at Bistineau was about 33 degrees Celsius. We know from previous work that people have conducted, it has been identified that when water temperatures increase above 31, you have reduced egg laying by the weevil. I haven't yet found any research that talks about larval mortality or adult mortality at higher temperatures. But as you can see, this site, Bistineau, had much higher temperatures than all the other lakes, and this continued from mid-June and didn't get below, even 30 degrees Celsius until mid-August. So a very long sustained period of very warm water. And these water temperature readings were coming from the top few inches of water, which is where the weevils spend most of their time in larval stage, egg stage, adult stage. So we suspect that at Bistineau, we have a couple years of all this data, that not only were the weevils having reduced egg laying, if they did lay eggs, I'm not sure that they hatched. We didn't see any signs of larval survival or that the adults were able to persist on this reservoir. And in comparison to most of the reservoirs or the water bodies that we work on, our sites at Bistineau were very thick mats of tertiary large giant Salvinia that was growing in full sun. And so it appears that the Salvinia just created this thick, blanket over the water surface with the full sun and was basically kind of superheating underneath that mat. So our factor that was limiting success during the early days was temperature extremes, both high temperatures at some of our sites, such as Bistineau, and low temperatures during winter in northern Louisiana. So I'm going to switch over and talk about a site that we worked at in central Louisiana. So, we began this work in 2012, and in 2015, Louisiana Wildlife and Fisheries coupled with the Red River Waterway Commission, and they released 150,000 weevils onto Lake Ayat in central Louisiana. And they did not have, at that time, an active monitoring program set up, so they really just released the weevils and sat back, to see what would happen. Well, they visited the site in February of 2017 and quickly noticed that the Salvinia just didn't look quite right. And they started picking up handfuls of Salvinia, and we're finding weevils lake-wide all over the lake, and very obvious. You could just pick up a handful of Salvinia and find some weevils in it. So they called me and said, we need to get out there, we need to collect some data and see what's going on. And for us, this was very exciting because we've been working on these lakes since 2012, and we had never really seen before, a self-sustaining, large-scale, lake-wide population of Salvinia weevils. So we developed 20 stations on this lake, and we decided to sample every 2 weeks for as long as possible. This lake does have a water level drawdown that happens almost every year, in part for timber harvesting. And so we knew the drawdown was coming. We started sampling, and we were able to sample from February 15th to July 5. And by the 5th of July, we had 9 of our stations that were inaccessible due to the water levels dropping down too much. So on this graph, you can see on this map, you can see all 20 stations, and the ones in the center here colored in blue are the 9 stations that we were able to consistently sample for the entire 5-month period. So we found some really interesting results. With this study, we were able to ground truth some observations and population dynamics that I had witnessed in my culture boxes for many years, but with the culture boxes being a slightly, at best, artificial growing environment, it was nice to see these same trends out in the real world with an established population of weevils. So for the population dynamics, what we saw was that at the beginning of the season, you've got these overwintered adults. So this is our sampling in mid-February, and we have quite a large amount of adults. We have 80 adults per kilogram of giant Salvinia at this point. But you have this inverse relationship where you have this flow from mid-February to mid-April, this decrease in your adult densities. So what we detected that for the most part, these adults that stuck it out and made it through winter basically survived winter, they mated, and then soon after died off after they laid their eggs. So you see an inverse relationship where the adults are dying off and the larvae are now peaking early in the season. Then as we enter more of just the warm part of the growing season, we see these sub-equal densities of the adults and larvae. So we're looking at this from a temperature perspective. like I said before, there was a lot of information done, a lot of work done in the 80s, mostly by Australian scientists that were initially working with giant Salvinia and the Salvinia biocontrol. And they used growth chamber studies with constant water temperatures to determine some of the low temperature limits of the Salvinia weevils. And from this data, the low temperature limits were very mild, so no larval survival at water temperatures of less than 17 degrees, which is only 63 degrees Fahrenheit. But what we were able to detect with this study is we had wondered for years, when you compare a growth chamber study to what's happening in the real world, where you have these naturally fluctuating, diurnal temperature regimes. And so through this study, we're actually able to detect weevil mating, egg laying, and egg hatch, and of course larvae at a lower temperature with temperatures ranging from lows of 14.5 degrees Celsius to highs of 17. So we're able to take this data and at least expand it a little bit, from previously thinking that eggs couldn't hatch at 19 degrees. We've now got eggs hatching between 14 to 17 degrees Celsius. And then from this data, we were able to use this as a threshold for future work in Louisiana when we were looking at winter temperatures and what we would expect our success to be with our Salvinia weevils. We also looked at nitrogen during this entire time that we were sampling on Ayat Reservoir. And we had a lot of previous work, again, in the 1980s by Australian scientists about how nitrogen levels affect Salvinia weevils. And we knew from this work that when a plant was more nutritious, had more nitrogen, that your larvae would develop quicker, the females would produce more eggs, and then you would overall get a higher weevil growth rate. We also knew that from room studies that at about 3% dry weight nitrogen is considered optimum for a good, chance at biocontrol success, weevil establishment, population growth. And then we conducted a few field studies in Louisiana where we detected that when you had plants that were less than 1.5% dry weight nitrogen, that you had reduced weevil densities and limited establishment at those sites. So it was interesting when at IAT we sampled nitrogen at all of our sites, all of our plants throughout this entire 5-month time frame, and we saw that nitrogen only varied between 1.5 to 1.9% dry weight. So it was interesting because the plants were above or at this 1.5%, but not much above it. So it really kind of took a lot of our worries away about if we were going to encounter many sites in Louisiana where nitrogen would be our limiting factor, because it looks like you really don't need a ton of nitrogen to have a sustained and supported population of weevils. So for the final part of this biocontrol talk, I'm going to talk about a success story from central Louisiana where we had consistent overwintering. This is Chico Lake, central Louisiana, and specifically our site titled Bridge Site. We went to Bridge Site in 2017, and we released 93,000 weevils and had successful overwintering the first year. We went back in the second year, and we went ahead and bumped up with another 94,000 Salvinia weevils, and again, we had successful overwintering. And so this is already similar to the patterns that I was just talking about at Lake Ayat, where they had successful overwintering for 2 years, and then the third year, we were able to see, large-scale weevil expansion. So for the third year, we decided, since we'd had 2 years of releases and 2 years of overwintering success, that we would not release any weevils. And this year, we actually achieved successful management of giant Salvinia at this site. So kind of the age-old question that everyone wants to know when dealing with biocontrol in general, and especially with the Salvinia weevils, is how many weevils do you need and how long does it take to manage Salvinia? So if we use this Chico Bridge site just as an example, so during the third year, after 2 years of successful overwintering, we had an early season weevil population explosion. So I sampled many sites all over Louisiana, different water bodies from central to south Louisiana, but we're just going to talk about Chico Bridge here, our red circle. So during this third year, from May to June, so from the blue bar to the gold bar, and this is adult weevil densities, we had an 8-fold increase in adult weevil densities from approximately 7 adults to 54 adults per kilogram of Salvinia. This value remained stable at about 60 adults in July, and then in August, we peaked at 140 adults per kilogram. And this August value was greater than all other sampled sites by at least 4-fold. When we look at the larval weevil densities, Our larval densities also peaked in August at over 100 larval weevils per kilogram of giant Salvinia. And moving into our dry weight biomass, at this site, Chico Bridge, when we started the season in May, Chico Bridge, and this is May of the third year, had greater dry weight biomass of Salvinia than any of our other sites. Then the weevils peaked from June to August, and from July to August, we had a 94% reduction in our giant Salvinia biomass at this site. And then from August to October, this site, which initially had more biomass than any other site, now has the least biomass of all sites. And percent cover, you would expect that to follow a very similar trend as, dry weight biomass. So with percent cover at this site, from July to August, we reduced by 88%. And then from August to October, the percent cover at this site of giant Salvinia only ranged from 8 to 10%. So back to our question of how many weevils do you need and how long does it take? So really, if we just use Chico Bridge as an example, at Chico Bridge we had 2 years of successful overwintering. This was followed by an early season spike in adult weevil densities. These high weevil densities were sustained for 2 weeks through June, from June through July, and then soon after that we crashed the Salvinia. So we still, when people ask us this, we cannot give a hard and fast answer of a true recipe, but we do know that weevil overwintering is the first key to management success. You are not going to have successful biocontrol of giant Salvinia if you do not have long-term establishment of the weevils from year to year. And I'm pretty much, I believe, out of time, and so I didn't have time to go into this. I just wanted to throw out here that this integrated management, that when I first started, working in Louisiana and working with biocontrol agents 10 years ago, I would have been very scared of herbicide treatments at my plots. I would have thought that that would, wreck my biocontrol work, kill too many of the plants too quick, and the weevils wouldn't become established. And I was also very nervous about lake-wide water level drawdowns, about that, taking away weevil habitat. But we have seen over the years, shockingly, that the Salvinia weevils respond very, very well to lake-wide water level drawdowns. At times it appears that sometimes it helps to bolster the population, that it brings all the plants together, the weevils, mate, reproduce, the waters come back up, and we're able to spread the weevils to further parts of the reservoir or the water body. Same with herbicide treatments. We're seeing more and more that we can sample a site that has weevils before herbicide treatment, and after the herbicide treatment, we'll come back a few weeks later when we've had some regrowth, and we'll see that basically the weevils have moved off the damaged plants or the dying herbicide-damaged plants, are moving on to any of the new green growth and are still persisting at the site. So I think there's a lot of potential for integration of multiple management practices for giant Salvinia. So just funding was provided by Louisiana Department of Wildlife and Fisheries and the Army Corps of Engineers. And I just want to acknowledge a couple of people from Louisiana that have, been on all these boat trips with me and sampled all these plants side by side, and then also some of the technicians back in our facility in Louisville that have helped make this happen. So thanks.
Thanks, Julie. Now we get to hear from Dr. Rodrigo Diaz.
Great. Thank you, Julie, and thanks, Chris, for those great presentations. So yes, I'm going to continue with this conversation about biocontrol and what we're doing here at LSU. So we have this awesome background already provided by the speakers. So I want to recognize right up front, that some of the topics that we're going to cover now are in a great team effort between Louisiana Wildlife and Fisheries, the U.S. Army Corps, and our cooperators in Argentina. That is basically the FUEDE, that is this awesome group working with biocontrol overseas. Let's start. Let's see. Page down. Awesome. So I think the theme is consistent. We're fighting against a rapidly growing plant. And then we have this issue between, predator-prey interactions where, the prey is growing so fast, and we are always playing catching up with the weevils. So I like this picture a lot because, it just highlights, how rapid it grows and the massive areas that we're going to be talking about. So remember that conversation. It's always we're fighting against this rapid growth of giant Salvinia. Now, Julie provided awesome background about the biocontrol of the weevil. This is just a close-up pictures And I tell my students and the people that I work with, that every single dot that you see in those weevils are made out of gold. So they are quite valuable, and I think they are playing a fantastic job in South Louisiana. So native from Brazil, and as you understand, Brazil is a tropical country, so you already can understand, some of the issues that we're going to be talking regarding this tropical weevil trying to make it in temperate regions of the Adventive range. So fantastic work by this weevil, and Julie mentioned some of the success, and this is terrific. So here in these pictures, you can see one of the most amazing examples that I have seen about how rapid biocontrol works with the Salvinia weevil. So this is an area in Cameron Parish, southwestern part of the state. So we went in June, and you see the picture of the Salvinia on the left. You see the coloration is already browning. And then we start collecting samples over time, and what it was amazing, it was just to see these tons and tons of Salvinia deteriorating and sinking in a matter of a very few months, as you can see. So as anyone working with giant Salvinia, by May, June, you are really concerned when you see those massive amounts early in the season. But our experience has been in South Louisiana that once you have successful overwinter of weevils, as Julie mentioned, you do have these great chances of the weevils being actually spread spatially everywhere. So if you take a look at that picture in June and you are in the boat with me, you will be grabbing one plant. I can almost assure you that every single plant that you see in June will have at least 2 weevils on those plants. So spatially, they were everywhere in June. And then the numbers increased tremendously, and you can imagine it just takes a few months for the plant to deteriorate. Of course, we're in the middle of the growing season. There is a heat involved. You suspect that the plant is under severe stress by the larvae, the tunneling effect, and also those feeding. But you wonder, whether there is actually bacteria and other pathogens finishing the plant, and then eventually it sinks and goes under. So it is a really amazing process, and you can just think about, if you're a wildlife ecologist, thinking about all these tons and tons of biomass sinking on those wetlands. And that's quite interesting in terms of, the ecology of those wetlands. Here's one of the most beautiful pictures that you can see in South Louisiana. It's just brown sinking Salvinia. So I think it's just fascinating to see, if I speak with someone talking about water quality, you will see that some of these wetlands, the water quality specifically, dissolved oxygen will be low, and you may even see fish kills. So again, every time that I think about this system, it's amazing to think how fast it can work in the south of the state, the rapid ecological changes that these wetlands are going through. So keep that in mind in terms of, what is going on in the south of Louisiana. Here I have another picture that is actually in Cameron Parish. As you know, two hurricanes went through this region, and all of this is probably gone. All of this will be, piles of Phragmites. But this picture was taken, 2 months ago. And what you see is, first, no forest, and definitely this is, beautiful marsh, coastal marshes. You see the color of Salvinia, that reddish, brownish, and some black. So that's just beautiful, just arriving in these massive areas and just seeing this variation in color. And as you can imagine, we have been using with Glenn Sur from the U.S. Army Corps of Engineers satellite tiles to measure NDVI and other plant quality features to assess on a larger scale these changes in coloration which are associated with plant quality. So yeah, it's a fantastic system and works extremely fast in the southern parts of the state. Now, okay, I just saw the message, sorry, from Courtney, so I'm going to continue. So something important that I have to follow up is that, in LSU, we're partnering with Louisiana Wildlife and Fisheries and CPRA, which are state agencies, and we are responsible for producing Salvinia weevils, similar as Julie, but we use these open ponds to mass produce Salvinia weevils, okay? So the advantages of these open ponds is that they have, Salvinia grows extremely fast, and you have these immense light source. Of course, they are fully open, and Salvinia grows tremendously, and all these leads to lots and lots of biomass that you can produce. And basically, by volume, you have higher amount of Salvinia weevils produced for a given area. Something very important that I want to highlight about these ponds is that we need to be located in the southern part of the state, because as Julie mentioned, the risk of having these open ponds in the north is just, we cannot afford losing all of this because of a harsh winter. So therefore, our operations are located south of Lafayette and south of Baton Rouge, specifically in San Gabriel. So this is our operation. We maintain a quality control system. We fertilize the plants. We check densities almost every 2 weeks. And then something important, just because, we are in South Louisiana, that means that, we are susceptible also sometimes to harsh cold fronts. This means that during the upcoming weeks, we are going to move some of these cattle tanks in the greenhouse just to make sure that we have basically we can be protected against the risk of a cold front. So this is just as a safe operation, right, moving the colonies indoors. So here is just a picture of the greenhouse with some of the team members of the Salvinia here in LSU. And here, so the idea is what we do is, we invite several stakeholders, including the Louisiana Wildlife Fisheries and several private landowners in the central and southern part of the state, and they come to these ponds, and they bring plastic totes, and then you see the boat in the picture, and basically we load these totes with Salvinia infested with weevils. And sometimes we have densities up to between, we try to harvest the ponds when we reach 30 weevils per kilo, but the densities vary drastically. So in every harvest, we do a quality control, and then we give them what is the density at the harvest date. And then the people will take these totes to the destinations. Usually, sometimes they drive a couple of hours to our locations, and they go back and release these weevils where they are needed, right? And something important we have tried to mention is that the way that you release these weevils is quite important. If you just dump the Salvinia on top of the floating mat, chances are like the Salvinia will dry and the weevils will not establish properly. I would like to mention that we also are helping other states sending Salvinia weevils. In this case, we extract the weevils and we send them, in a small containers. So we have done that with, as Dr. Mudge mentioned, with Santee Cooper in South Carolina. We will send to Mississippi, and Florida we send the common Salvinia weevil to help them. So that's another biotype of the Salvinia weevil. So we help Florida, Texas. I got a chance to go and help also Puerto Rico. There is a urban lake, heavily infested in Puerto Rico, and also in Africa with Cameroon and South Africa, where despite that they have the Salvinia weevil there, seems to be that their colonies were infested with a pathogen. The weevil had a pathogen, so we sent them the weevil that we tested, and we didn't have that pathogen. So it has been fantastic, literally trying to assist other regions with the population that we currently have in Louisiana. All right. Also, what we do at LSU, we do have an extension component in the program. And thanks to, I want to recognize, of course, Dr. Mudge, who has been helping us, promoting and creating these material for the public. For example, about, what IPM strategies are available to manage Salvinia. So here is just, I wanted to put some some publications that we have done about the Salvinia weevil, how to recognize the weevil, what we can do during the winter, and how to produce and release them. So we have all of these is available in the Giant Salvinia website. And because I know with Chris, we are bombarded sometimes by the public asking, so this Salvinia website has been very helpful to direct the people for access to more information. Something really interesting is that, lately the Ag Center has been helping us with social media. And I want to mention here that, we can reach a vast audience just by creating these materials, for example, for Facebook posts, Instagram, as well as Twitter. So thanks to a fantastic team of extension agents and people in the Ag Center, we have been able to post and share this information to more audience. And I want to recognize something really cool that my technician, Corey Pham, developed. So she created an Instagram page for our program, and it's pretty cool. It's called We Will Rock You, and I think it was a pretty awesome name that she came up with. So, and the idea is, she posts pictures about activities, what we do in the ponds, how we go about sampling. So it's really fun. It's a really nice way to interact with the public. Again, our goal is to spread the word that, the Salvinia weevil helps, provided that, we give them the right environment. So anyway, so this is our effort to spread the word about the weevil. Now, I'm just going to, in the last part, talk a little bit about the research that we're doing. And what we just think about biocontrol, I like to think that, this is perhaps one of the most cost-effective approaches of methods of control. So provided, as Julie mentioned and the previous speakers mentioned, provided that we have good overwintering and those populations are self-sustaining. So we're going to talk a little bit more about that. This is a picture by Jeff Sibley, who works for Louisiana Wildlife and Fisheries. And this is what we're fighting in terms of issues in the north of Louisiana. Sometimes we have harsh winter, and the weevil from Brazil may not be exposed to those winter temperatures in the native range, so they have a hard time. So as you can see, we can form ice. I don't know what's the difference between the cold tolerance of the plant and the weevil. I suspect that it's microclimate that protects the plant, but I think both of them may be susceptible to cold. It just happens that the plant may create and find a microclimate. So we might discuss that with Dr. Moja about, who is more tolerant than who. But yeah, this is what we're fighting against, right, in the north. How you survive under these sometimes harsh conditions. So we did I just wanted to mention here that we did this comparative work, sampling populations in the north of Louisiana as well as in San Gabriel, right, in these open conditions. So we did this using Berlizzi funnels, which is an awesome tool that we have to assess weevil densities. And then we also, from the females that came out of those samples, we did a dissection to determine, which one is reproductive just by looking at the presence of immature eggs in the ovarioles, as you can see in that picture on the right-hand side on the top. And these are some results. So in general, you find higher densities of adults in San Gabriel. This is, week number 1, that would be January, all the way to May. And what you see, the population in San Gabriel, South Central Louisiana, is higher over time. You see the population from Cross Lake, it starts decreasing in January, February. We have weevils, but you start seeing the similar trend as Julie presented by May, how these two populations, these two conditions, the densities start separating, the north going down. In terms of the females that are reproductive, so when you dissect those females, the bar, you see the San Gabriel, the black bar, you see a high percent of females are reproductive in January compared to Cross Lake in the north, right? But notice that, by February, you start seeing some females, at least they are reproductive in Cross Lake. So that the story starts becoming more interesting. And then over time, you see in March, females are reproductive in the north of Louisiana. And this is very interesting, and for me, it's very telling. So this is the larvae, okay? So you see the larvae with black, that's San Gabriel, Central South, and then you see the pretty almost absent January, February, and then, in San Gabriel you start finding larvae on the plants, and then, but notice the gap that starts forming with the northern population. You start coming, week 12, week 13, and really minimal numbers of larvae. And then by week 15 is when you start seeing more. So, and I think this delay in the arrival of the larvae seems to be that create these, is the beginning of, is one of the explanations of why in the north of Louisiana we have so much issues. Because, as Julie presented before, there is not much adults replacing those that die because of the overwintering. So yes, the females were reproductive. They were capable of laying eggs. maybe they lay the eggs at those temperatures, but maybe the larvae is just not handling those temperatures, and that's why you start seeing those numbers back there in terms of those delays. So again, it's just a fascinating system about how the delay and the issues in the north start coming to being. So we have tried several approaches about how we increase the microclimate of the winter, and here we try with some evaluating fabrics that we can put, you see those kiddie pools, and the idea will be to raise the temperature so in the winter we have better survival. We also try to increase the Salvinia, the temperature, by crowding the Salvinia, so we raise the temperature just by having, thicker mats. And here are some pictures that we did and some of these research that we did in North Central and South Louisiana with different thickness of Salvinia. So again, trying to improve the survival of the weevil. And here in the last part is basically I want to highlight this other approach that we're trying. Is basically we're trying to look for a population of the weevil from most southern locations, that will be Argentina, right? Going to the native range and finding a more cold-tolerant population of the Salvinia weevil, okay? So we went to Argentina, and you see the latitude between 33 and 34 degrees south, and as you can imagine, the temperature in those regions matches more to Central and North Louisiana and Texas, right? So we published this information about, how this weevil population is the same species, right? How is basically the same species. We use morphology and genetics to recognize this, and also we compare several variables about cold tolerance. So when you put the weevil from Louisiana next to the weevil from Argentina, you do notice that the weevil from Argentina actually recovers faster from a cold exposure. It's 1.8 times faster. They wake up early after 0 degrees centigrade. And then in terms of survival, so as you see, they basically at the same exposure time, the weevils from Argentina actually had a higher survival. In this case, was at 0 degrees centigrade, right? And all of this is just trying to say, okay, what this population is going to bring if we release it in Louisiana, right? So in this case, we show, yes, it is a bit more cold tolerant and can help, right? Of course, we cannot measure every single variable of these weevils, but hopefully with the new project that we have with the U.S. Army Corps and Louisiana Wildlife Fisheries, we'll be able to characterize more with the end goal of releasing this population and working with Julie, for example, in establishing a mass breeding facility in Central and North regions. So we have active work going on right now with this cold-tolerant population. So that's awesome. So at the end, the final message is pretty straightforward. In terms of biocontrol, it's by far the most sustainable method of control of Salvinia. We always need to be fighting, especially I like what Dr. Mudge mentioned about the efforts that we need to do early in the season when you are not fighting a monster of 1 meter thick, that is the summer. So we need to move our strategies early to help the weevil, help the herbicides by managing the bottleneck that occurs early in the season. Mass rearing is important. You have seen flooding and other disturbance actions where you can, the weevils may be reduced. So we need to maintain these rearing operations to supply more weevils to these locations. And finally, research, as far as we know, is definitely we're trying to get how we improve the biocontrol program in northern regions. That's where we're putting a lot of effort. And with that, I want to thank you for your attention.
Wonderful. Thank you, Rodrigo and Julie and Chris. At this time, let's go ahead and take some questions. Tara is going to work through the chat questions for us, so if you can continue submitting your questions there, and she'll read those aloud for the presenters to respond to. Or alternatively, you can be unmuted to ask your question. So our event producer is going to give us instructions on that process.
Thank you, Courtney. To enter the question queue, please press #2 on your telephone keypad to enter the question queue. You will hear a notification when your line is unmuted. At that time, please state your name and question. Once again, pressing #2 will indicate that you wish to ask a question.
Great, thank you for that. So Tara, if you want to go ahead and walk us through the chat, that'd be great.
Yes. All right. Thank you very much. Our first set of questions, we have 3 questions for Dr. Mudge. And the first question is, when Salvinia is successfully managed, is there a pre-existing seed bank of other invasive weeds poised to reassert their presence?
Yes, well, it kind of depends. If we see an infestation of giant Salvinia that's only been there maybe 1 or 2 years, I think that whether it's a native or another invasive, it has an opportunity to come back, especially if it's an emergent. But once we start to get prolonged periods of time, the light, the oxygen, the pH alteration, all that stuff has occurred. Rodrigo and I are both on a committee. One of his students is actually looking at submerged vegetation like coontail and kabamba, and looking at, how does that plant survive, even like insect populations, what's the change over time in that. So we definitely see an alteration, and they can reassert themselves if it's a short period of time, such as where Rodrigo showed that site in southwest Louisiana in Cameron, where the Salvinia wasn't there for long enough and started to see some other vegetation popping up. But if it's probably something like a cattail, an alligator weed, or something else that can stick itself up and above the giant Salvinia, it's going to have a better chance of something that's at water level or below.
All right. Thank you very much. Our second question for you, the respondent noted that giant Salvinia sounds similar to kudzu. If we continue to have mild winters in the southeast, it seems like the only areas that will have reduced Salvinia coverings are the places that have been chemically treated. What are your thoughts on this?
Yeah, it's definitely aggressive like the kudzu. The one thing Dr. Mike Netherland asked me years ago, he said, well, if Salvinia is so bad, why don't we see it everywhere in the southeast? And I think a lot of that was, I couldn't answer it, even though I should have known the answer right away. It took some just experimentation and going around and about. And we found that the pH in the water and the system was a little high, so places where you see hydrilla and other submerged vegetation, those places are not conducive to expansion of giant Salvinia. So the southeast in general has some great places for it to take off, but if the pH is low enough initially, it's not going to be able to take over. Chemical treating will be it. If it's initial infestation, you want to do a rapid deployment to try to control it through herbicides. That is, but once you get in places like Ross Barnett and the Santee Cooper system, if you don't have a good feel for where it's at, the weevil will also be an opportunity, depending on the winter conditions, to kind of get it out there and keep it in check when you can. So it kind of depends upon, herbicide is going to have a big part, and even if it's a small infestation, rapid deployment of whatever you have, will be key.
All right, thank you very much. And our final question for you, is giant Salvinia primarily being spread by boaters, moving a boat from one water body to another, and birds?
The wildlife is probably pretty minimal. It's a little bit easier to pick up something like a hydrilla or a duckweed by accidental or purposeful movement. The Salvinia, if it gets a decent amount of size, and I don't know how much, we haven't really traced it. I would say the boats are a big part of it, especially early on from the Toledo Bend days in 1998, 1999. We saw that it gets in between the boat and the padding on the trailer. And former Ag Center employee, Dr. Darrell Sanders, had noted that for maybe up to 2 weeks they were finding it almost like sponge-like and surviving in that cool, moist area, being like a sponge. And even some people talked about putting on the ground for a day or two and it coming back. So partial boat, some of it, a lot of it is our systems are connected. So if you start in North Louisiana and there's a lot of flowing water that moves from one to the next to the next, so it can make its way down eventually from the northern part of the state until the south, until it basically reaches the really salty, brackish areas where it eventually will die. So it's a little bit of everything, best we know.
All right. Thank you very much. Our next question is for Ms. Natribe. Have you found any negative effects of the Salvinia beetle? Are they outcompeting any native insects for resources?
No. With the Salvinia weevils, they are completely host-specific, and they are, feeding on the plant that they evolved on, from South America, from areas like Brazil and Argentina. So the whole, idea of this is that, the Salvinia weevil has evolved to feed on giant Salvinia, and we have giant Salvinia in the U.S. now, so we import, its host-specific insects. We do see, some caterpillars that are generalist feeders that will actually try to outcompete the Salvinia weevils because caterpillars can be such vigorous feeders. And so we'll see some of that, and some of these caterpillars actually were originally from other countries and snuck in with, plants like water lettuce, some other aquatic invasives. So, no, the Salvinia weevils are definitely not, outcompeting or taking up space that other insects could utilize. I wouldn't say so.
All right, thank you. And I think you touched on the final question that we have in the chat right now, but I'll still reiterate that. The question asks, are the weevils exclusively feeding on giant Salvinia, or will they also affect common Salvinia?
So that's a tricky question, but yes, they can feed on common Salvinia. Rodrigo might want to pipe up in a second and see what he thinks about this, but the current information is that there are two distinct ecotypes of the weevil. So they are the same species. They are all Certivagas salviniae, but there is an ecotype that specializes on giant Salvinia and an ecotype that we primarily see in Florida that specializes on common Salvinia. But quite a few years ago, they did genetic analysis of, the weevils that they located in Florida feeding on common Salvinia versus the weevils feeding on giant Salvinia, and the DNA was not distinct enough to categorize it as a distinct species.
That was great. Yeah, just follow up a little bit with Julie. So that was on target. I just wanted to mention that, we have been sending common Salvinia weevils to different locations. We have the common Salvinia biotype, let's call it, or ecotype, well established in Central and South Louisiana. We have it, for example, in John Lafitte, that is west of New Orleans, that is doing a great job at controlling common. So, yes, and then we have it all along the Maurepas region. So sometimes we have stakeholders asking, what is going on with the common Salvinia weevils, whether we rear them, and we just specify the LSU only rears the giant Salvinia ecotype, but we do know about these wild populations nearby. So what we do is usually we go to the site, do an extraction, and if someone needs them, we confirm the identification, and then we direct them to where the common Salvinia weevil is. So we try to help at least with access to these agents.
If I could chime in one follow-up thing, also plant identification is proper to know which one you have, because especially from the weevil rearing standpoint, Rodrigo and Julie are, the weevils are being spread throughout the states, and they're being, released and contracted out. But if you have a common Salvinia site and you go in and release the giant, because you're not like some of these agents where you're releasing just the insect, because you have to bring in the host together. So you could actually infest giant Salvinia in a common, or you may do vice versa, and you have the common, giant and Salvinia in common, and then all of a sudden you go from not being able to reproduce because the common can, like Julie and Rodrigo said, there's host specificity and feeding, but then reproduction and all that. So just proper plant identification, and Rodrigo and I have on the giant Salvinia page he started, and I kind of jumped on board with that. We put in a little common and giant identification. He has the videos, and then I have some chemical recommendations on there. So actually we're trying to make sure everyone knows from the start where do you stand from, educationally speaking, before you go and accidentally release the wrong thing.
All right, thank you for adding that in. We have one more question that came into the chat, and this can be for really any panelist, please. Giant Salvinia is listed as a state noxious weed in Colorado, Oklahoma, Maryland, and others. There are no reported accounts in Nebraska. What might be the reason that giant Salvinia has not yet been found in this state?
Julie.
So I'm thinking, I would suspect that giant Salvinia is not Nebraska, maybe that it can't overwinter. A lot of these a lot of these states that you may see listed on the larger map of, the 13 to 15 states in the U.S. that have had infestations of giant Salvinia, a lot of times these are not sustained or reoccurring infestations of giant Salvinia because, if they get cold enough winters, it knocks it back. So that would be my guess with Nebraska, that, even if a few pieces, snuck over there on someone's boat trailer and got to Nebraska, that, that winter would take it out probably.
Fantastic, Julie. Sorry to put you in the spot, but that was on target. Just to follow up and expanding on all these winter issues, we do have a problem already with southern Arkansas. So if we have this as a harsh winter, all those populations may be knocked down farther south. So, yeah, winter dictates tremendously the management that we're going to have. So every year is different. The 2 last winters were mild, so we have in the south awesome success of biocontrol. But this winter may be different. If we have a cold front coming, as we mentioned before, maybe 2 or 3 of them, we can seriously be thinking about reduction of Salvinia as well as weevil. So yes, the upcoming months are very interesting as the managers of the program, like how we are going to handle the subsequent year.
All right. Well, thank you very much, everybody, for providing that answer. That is all the questions that we have in our chat, and we are approaching our hour-and-a-half time limit for the webinar. So if anybody else has any additional questions, please do feel free to note that in the chat, and we can provide those answers then via email to make sure that everybody receives those. Thank you, everybody, for participating. And, Courtney, I'll turn it back over to you.
All right, thanks, Tara. And our event producer confirms that there were no questions waiting in the unmute queue, so we'll go ahead and wrap up. But again, feel free to reach out to our presenters. Chris, Julie, and Rodrigo, thank you guys for taking the time to share with us today and share your work and your program. Do you guys have any parting thoughts for us? We can start with Chris.
Thank you, obviously. And one thing I always hate to say is I hope giant Salvinia spreads to other places, but that's one of the unfortunate ways I think we're going to get more exposure. Maybe we can put it somewhere in Washington, D.C. or some big national spot in the Washington Monument around there, the National Mall, just for a few months. But all kidding aside, it would be nice to get some more public understanding of what we're doing. We're trying our best, and it's a battle. We all enjoy it, and we're trying to do more collaboration between different parties, and it's job security. So, and thank you for your time.
Thanks, Julie. Do you have anything else for us?
Yeah, no, just thanks for having me, and I agree with Chris that a lot of times it's hard to get across to policymakers and other people, what we're going through, and that giant Salvinia is an amazing plant. It's very tough. It's very persistent in our reservoirs, and we're just going to have to keep coming at it because it seems as if it's here to stay in most of our, warmer regions. So, thanks.
All right, thanks. And then Rodrigo, any final thoughts from you?
Yeah, just pretty quickly. So thank you, everyone. Just basically that we need to think about the economics behind all of this. I'm an entomologist. I think about insects and sometimes weeds, but bringing an economist into the conversation, seeing, how the investment that we're doing, how this management action leads to eventually reducing the cost of control and the returns. So I look forward to see, the economic impact of our programs because it will show that how research and education brings, a more effective management program, especially if it is translated in reduction of cost for our stakeholders. Thank you.
All right. Thank you, guys. I do want to call your attention to the chat box one more time. I posted a link to a short 2-minute survey to request your feedback on our presentation today. And then as a reward for completing the survey, Dr. Diaz has shared the link to their Instagram account, the Weevils Rock You. So go ahead and check that out as well. And then lastly, I want to mention next week's presentation at the same time using the same WebEx process. We're going to learn about concepts and strategies for emergent plant management by Dr. Stephen Inlow from the University of Florida. So please watch for an Outlook invitation announcing those details. And with that, I think we're done for the day. Thank you all for tuning in. I hope you have a great afternoon, and I'll look forward to learning with you again soon. And that will conclude our meeting.
Thank you for using event conferencing. You may now disconnect.